so sorry. Is that on?
Yeah.
Oh.
Okay, one more time.
I have a question.
All right. So I'm going to go through the beginning, through, and then we're going to ask questions. Okay. So you have a 6-foot long piece of DNA in your body. Each of your cells actually has 2 of them. One from your mom and one from your dad. Those pieces are actually broken up into 23 smaller pieces called chromosomes. During synthesis, each of those pieces gets copied. So we're just looking at one genome here.
She's like right there.
No, I know, it's good camera. It's a good camera work. So there's the original strand and the copied strand. If this were a T, this would be an A. If this were an A, this would be a T. These 2 things are exactly the same. So if this was from mom, this is a copy. Same thing. Now, during mitosis, these condense, which means they're wrapped really tightly around histones.
You can't see it on the camera, that's why.
One other thing to remember is that after they're copied, they're actually connected.
It's gonna be a very shaky video.
Okay? They're actually connected. So, in the end, you have a copy, and together, here you have 2 genomes. Now, if you look at one piece, one chromosome, which is now actually 2 copies, which we call sister chromatids, there are a layer of proteins actually ringing or around a centerpiece of that chromosome. Those proteins together are called kinetochores. They attach to motors, and in the end, they're really, really important because they're going to stabilize microtubules. Okay. Now, don't forget that we have an entire other genome to which the same thing is happening. It would line up here in the middle of the cell too, and we would pull them apart so that each cell has a copy. Right? You would want each cell to have the entire genome. So this is a copy and this is a copy, which means... All right, thumbs. That is a bold-faced lie, people. You do not understand it that well.
Yeah.
Ask me some questions, Canny.
Okay.
In English.
What
No,
else
you
would
know
I
what?
speak?
Think about your question and then ask it again.
Can you go to the part of the
Sure, kinetochore.
Yeah.
Okay, so you have double-stranded piece of DNA. Double-stranded piece of DNA. There's a ring of proteins that wraps around them. That's called the kinetochore.
Okay.
The region of the protein that it wraps around is called the centromer.
Is the kinetochore already there, or does it?
The kinetochore assembles.
Once the 2 genomes, or once it's, where are the 2 genomes when the kinetochore assembles?
All right, so I think there's a little bit of argument here, but
So when they go to the
there's
nucleus-
probably a portion of the kinetochore that is actually here.
Okay.
So technically, if you have a double-stranded piece of DNA and you were to copy it, what would actually happen is there's a loop of DNA or a loop of protein right there, and when you copy it,
So it's just naturally made around it, kind of?
yes, ish. Let's not ask too many questions about
And
that.
then the microtubules grow and then connect to them.
The microtubules assemble.
Well, they will try going back and forth until they finally.
They will not try to do anything. They will assemble and they will fall apart
Until they hit the.
until they are
chromosome.
stabilized by either a kinetochore
Or the-
or another microtubule.
Okay, and that's when they're getting ready to rip them apart.
Because technically, these little guys have little proteins on them.
So attachment.
Yeah.
How does it go from the histone to the condensed chromosome?
Okay, so in order to copy all of this, it has to be slightly unraveled,
Okay.
right? And in order for you to read it, it would have to be unraveled. If you wrap all of it around small little beads and then wrap those things together, So have you ever taken like a shoelace and started to spin it, and first it will form a spiral, and then it will kind of fold in on itself, and then it'll kind of become a glob? That's basically what's happening here.
Don't you have topoisomerase?
And that happens during mitosis, like during the actual clip.
Once the M cyclin is activated and condensin is activated, this will all condense,
Okay.
right?
Okay, yeah.
Starbuck?
When does that happen? Does that happen after?
Nope, too many pronouns.
oh.
Go.
What's mama do? Or what's
All
mama
right,
mean?
so we're just talking about mom here. There is a whole nother genome
Oh, for
that is
dad.
doing the same thing
Okay.
Simultaneously?
simultaneously. As a matter of fact, it'll be distributed, if this is the center of a cell, there'll be other chromosomes here from dad.
Do you ever have a cell that, is it impossible to have a cell that only has one genome?
Your sex cells have one genome. Remember we said that you're going to give part of your mom's genome and part of your dad's genome to your child? So you're actually giving some of your mom's, literally your mom's DNA to your children. I don't like your mom very much, so.
How does the kinetic
That's
work?
gonna be on the video.
Hi, Mrs.
Better take it back.
Damaris.
Take it back.
Take it back.
I'm sending an email out.
How does the kinetic core recognize the sequence of the centromere if it's wrapped around?
That is a really, really good question and one that I can't answer.
It was
Okay. Can I tell you the other secret?
2-minute warning.
Yeah.
All right. Here's the other secret. The kinetochore, when it is unbound by a microtubule, is likely producing a signal. Here. Ooh. No? No.
No.
Producing a signal. Is everybody with me still?
Is there one centromere for each chromatid or each genome?
There are 26 centromeres per genome, one
Okay.
centromere per chromosome.
What's a centromere?
The region of DNA around the that the
So why are
kinetochore
there
should run.
26 if there are 23 chromosomes?
There are
Around?
46
46.
because there's this genome and dad's genome.
Or in a minute.
This should
So
Yeah.
there's 23 chromosomes here, 23 chromosomes.
Yeah.
That's 46.
Yeah, but you said something about 26.
No, there's nothing 26.
Mr. D, can we go over this again tomorrow?
Absolutely not. Never again.
Please?
No,
Pretty please?
I don't like you.
Mr. D, we're done.
Because our English teacher might call us if we're late.
Should our...
Yeah, go.